Okay, okay. So thank you all for waiting. We're terribly sorry for that. Um, a todos que vieram, eu peço desculpas pelo que aconteceu. A gente teve um pequeno imprevisto técnico aqui com a transmissão, mas acho que agora a gente conseguiu resolver. A apresentação está na tela. Então, refazendo as apresentações, hoje a gente vai estar recebendo, tendo a imensa honra de receber o professor Gavin Naylor, do Museu da Flórida, um exímio especialista em tubarões, vai estar falando para a gente hoje o título da palestra dele, por que temos tantas formas diferentes de vida no nosso planeta. Um detalhe importante, se vocês tiverem perguntas, por favor, usem o chat. Se tiver pergunta em português, pode fazer em português que eu traduzo a pergunta numa boa. Se tiver em inglês também pode fazer, não tem importância nenhuma. Tranquilo? E é isso. A gente pede desculpas mais uma vez. So I thank you all for being here again. We're terribly sorry for what happened. We have this technical issue with the broadcasting, but now I believe we're ready to go. And once again, we're having the honor to receive Professor Gavin Naylor from the Florida Museum, who is a very good shark specialist, very renowned one. And he's going to talk about why are there so many different forms of life. That's the title of his lecture. And on behalf of the organization committee, Mr. Gavin, thank you for being with us today. We're terribly sorry for this technical issue, and the word is yours.
Great. Well, thanks again. Let's hope the second time we have better luck. So thank you, everybody, for being patient with these technical hiccups. So as I was starting to say, a fundamental question for biology is why there are so many different kinds of organisms on the planet. And it's strange because they really don't have to be. You could imagine a similar planet with similar environment, similar thermal dynamics, distance from a star, that had life on it. And you can also imagine that there would just be one species, maybe a bacterium that would crust and sort of have chemical exchange and replicate on the surface of the planet. But it's not the case on our planet. We have spectacular range of diversity and in size. And I actually did some calculations, and it turns out that the range in size of organisms on our planet spans 28 orders of magnitude. That's 10 to the 28th differences in size. There's these fungi in the northwest of the United States, which weigh thousands of tons, all the way down to the viral replicants. So there's a huge variation in the size of these things and a huge variation in the diversity. There are 8.7 million described extant eukaryotes. That's just the eukaryotes. That's a tiny fraction of all of the life. And that's just the ones that are alive today. They're hugely diverse. We've got things like redwood trees on the one hand and microscopic Chlamydomonas on the other. We've got blue whales, which are mammals and weigh 100 tons. And then we've also got other mammals, so relatively closely related, bumblebee bats that weigh a couple of grams. So both are mammals. One flies and weighs a couple of grams. One's in the water and weighs 100 tons. They look completely different, but they're both relatively closely related in the big scheme of things. So this is remarkable. And I think the biologists sometimes forget to be amazed when you're in the middle of this beauty. You to look around. You forget to be amazed that we're not just looking at a rocky seascape. We have the place teeming, bristling with amazing life. And in Brazil, you have the Amazon, which is, the centerpiece of some of the world's most amazing diversity on this planet. So if life, all the different life forms on this planet come from a single origin, and we suspect strongly that they do, because they all, in spite of the diversity, they all have their DNA-based life forms, they have RNA and DNA, the same mechanisms and architecture for replication. So we think the easiest way to explain that is if it arose once. So if all of the diversity that we see around us stems from a common origin, and we've got things as ridiculously different as redwood trees and bumblebee bats and whales and birds and snakes and beetles, if they're that diverse and they all come from a common origin, then the process of becoming different, the process of acquiring traits, has got to be a fairly easy thing to do. If it was a hard thing to do, then there wouldn't be so much variation. And ever since I was a child, I grew up in Africa, I was always fascinated with this idea of how organisms acquired traits. And I used to like the stories by a writer called Rudyard Kipling, and he would write these just so stories, how the elephant got its trunk, how the leopard got its spots, and how the rhino got its skin. And he would have these very funny narratives, like the elephant got its trunk because it was curious, and it got its trunk stretched because it kept going down to the edge of the river, and a crocodile grabbed hold of its trunk. So this is a fundamental question that all humans have, usually young children's. Adults seem to, become more accepting, and they forget to keep asking these important questions. Well, now I'm a faculty member and a professor. I'm still interested in these same questions. The same motivation drives me. But we have different tools, the scientific method, and we try to explore scientific explanations for how animals become different. For example, I work on sharks, as Arthur mentioned. One thing that's very peculiar is how these hammerheads acquired their very peculiar expanded heads that are stretched out laterally. How did that happen? Well, we'd like to know the answers to these questions. So another way of asking this is there something intrinsic about the way living organisms are built, about their architecture, that predisposes them to sprout new characters, to diversify and to become different? To explain why our planet is not covered in a single organism, a single alga or a single bacterium. Is there something intrinsic about the dynamics of living systems that makes them diversified? And I think there is, and I'm going to talk about that today. Well, I work in a natural history museum, and you guys are based in a natural history museum. What we tend to do is document all the different forms of life. We see something new, we say, this is a different species. We write it up, document it, and say, this is different, that is different. We make a catalog of all the different things. Well, evolutionary biologists, and I'm also an evolutionary biologist, try to explain how these differences come about, and they look always to environmental influences to account for the diversity. And they say, natural selection has shaped this, that shaped the elephant's trunk, it shaped the bee's wings. So they look to these external forces that sculpt the phenotype of these animals. So always, you see these figures here that I'm showing, these wheels, and they show where selection has acted to cause the diversification of these animals. Well, I'm not so sure that this is a very fertile area to go down. I want to present you some ideas, which I think are a little bit different, but I just want to rock the boat, because students need to question everything. I mean everything. Don't just accept what your professors say. We need to challenge everything. That's how we improve. So to provide you with some context for this, back in the 1850s, when Charles Darwin and Wallace were exploring ideas about the origins of life and diversity, you have to remember that ideas, in order to be propagated, have to be transmitted in a way that people will understand them. They have to use analogs that people will grasp easily. And the prevailing culture at that time in the 1850s held that all of life had been sculpted by the creator. And each one was perfect because the creator was omniscient. And if he was going to make things, of course, he'd make them perfect. Nobody would want to say, you messed up here, this is not a very good design, send it back. So because it was made by the omniscient creator, all organisms were viewed as perfect. So also at this time, we were in the full swing of the Industrial Revolution, where people were making these machines that could replace humans to do automated tasks. They were unimaginably complicated. with thousands of different moving parts. And each of these different parts had a distinct function, which when put together collectively would do things like, make buttons or sort cotton or make huge swaths of material. So in the middle of when Darwin's ideas came around, we've got the Industrial Revolution in full swing, and we've got a prevalent view that life on Earth was created by divine intervention by the deity. So it was only natural that Darwin's new ideas would lean on these established concepts, or at least concepts that seemed credible, that people could say, yeah, it's a crazy idea, but I see where you're coming from. And those crazy ideas were that organisms were perfectly adapted to their environment. So we're not shaking rocking the boat too much. If the deity made everything perfect, we could say, yeah, organisms are perfectly adapted. That's true. And then we could also say, well, they were made like machines, the sum of their component parts. So both of these ideas, which are alive today, are holdover concepts from the divine creation, which made everything perfectly, and the Industrial Revolution, which treated organisms as machines. So for the people that thought that Darwin had a good idea, the baton passed really effortlessly from the creator, the deity, to natural selection. Everything was still perfect. It was just that the shaping of life had moved from divine to the sterile stage of economics and competition in a world of organismal machines. Well, Darwin was a very honest scientist and deeply curious and really wanted to get to the bottom of things. This was before the time when people just wanted to publish whatever they could publish to see what they could, so they could get promotion. He wanted to know. He wanted to drill down and get to the bottom of things. And he very fairly asserted that there were two components that were necessary for evolution to occur. There had to be some way to generate heritable variation, because organisms looked like their parents. So if the parent had a particular trait, there was a high probability that the offspring did too. And he advocated that if there's all this variation and you generate more offspring than there is a habitat to support them, there's going to be some struggle for existence. And some of the variants are going to do better than the others. So this was his idea of natural selection, which beautifully explains how some forms win out over others. But Darwin's theory did not provide a mechanism for the generation of variation. And people forget that. They just go, Darwin, natural selection. But he also asked this very important question. There had to be a way to generate traits, and those traits had to be heritable. So at the time, Darwin was criticized by many people for failing to provide a mechanism to account for the generation of variants. And he also was very worried about it. He said, yeah, you're right. It's a good point. I don't know. I don't know how this happens. Well, 100 years later, with the discovery of DNA, a mechanism became available. And people immediately seized on it. This is the thing that was missing. We now have DNA, codes for traits. DNA is heritable. So this explains that heritability, that the traits were coded for in the DNA mutates, and then the traits change, and that provides variation, and then natural selection works on that variation. And this would explain how the variation that you see in a population of birds here would be winnowed over time with new variants coming up into different species. So everybody really rejoiced that a mechanism was found to account for Darwin's conundrum. And this was the new synthesis. But the machinery generating variation held that a small change, here's got a little button here on the left. So you turn this little button, you rotate it to the right, and the genotype, you change the genotype, and then the phenotype changes too. So you can imagine as you flick this button, so this lever changes. Small changes in the genotype somehow, magically, bizarrely, gave rise to small changes in the phenotype. Well, you can see there's a problem with that, because What is the chance of some random mutation giving rise to a new viable phenotype? Remember that all of these organisms were perfect. They were built perfectly. And so if you've got something that's perfect and you change some part of it, it's presumably going to be less perfect. So we're asking DNA to have random mutations, and these random mutations give rise to new functional and operational phenotypes. The chance of that happening is extremely small because remember, these things are all perfect. And so a variation in that perfection is going to make it less good. So to get around this, the new synthesis advocated that evolutionary change was a series of very unlikely events. But those low likelihoods of something good happening were going to be offset by large numbers, huge numbers. So given enough time, given enough tries, billions of years, billions of organisms, populations expanding and contracting every generation, we can offset the low likelihood of novel, viable variants arising by chance. So this is the rescue clause. It's a little bit, I like to think of as a million monkeys and a million computers in a million years. One of them's going to come up with all of the sonnets of Shakespeare. So it's a sort of probabilistic argument. And that really is the basis of the way we view evolution. Modern biology still treats organisms as though they are built like machines, highly optimized systems of component parts. If you don't believe me, have a look at Wright's adaptive landscape, where you've got these regions, these peaks of adaptive optimality separated by troughs of where things are suboptimal. And you have to traverse from one peak to the other through the suboptimal peak or trough. If organisms were built like this, then any perturbations or mutations would almost always lead to component failure. They'd break. Novelties would be extremely rare, which would make diversification unlikely. So if we view organisms as machines, like a toaster, here's a toaster. You go and get some random toaster, throw in a couple of bolts, maybe put in a different circuit, pull off a knob. What's going to happen to your toaster? Well, chances are nothing. Or what might happen is it'll break and catch fire and your toaster is now broken. You can't just throw random things into your toaster and have it magically do something cool. It's not going to happen. This is not what we see in nature. Mutations don't break organisms. Some of them certainly do, but a large fraction of them result in the generation of new variants relatively easily. One of the organisms that Darwin was interested in was pigeons. If you look at a group of pigeons, there's huge phenotypic variation even within flocks. They've got different plumages, different attributes, and they arise repeatedly. So to use the toaster analogy, a living system, if it's a toaster, when we have variants arise randomly, what we see is the toaster doesn't break, but it may undergo replication. So we go from a 2 system to a 4-piece toaster. It may get a sort of more Germanic design to it, or a more Italian design with some colors in it. This happens. We see this. This is what happens in nature. And sometimes, not all the time, but sometimes in nature, this happens. The toaster turns into a vacuum cleaner. We can, through a slight change, we can transform into something very different. This happens not very often, a saltatory event. So the view that organisms are optimized machines sculpted by natural selection, in my view, is probably not the best lens through which to understand the generation of biodiversity. I view this as a 100-year-old intellectual cul-de-sac. I think that we need to start thinking about other ways that makes, that can be responsible for the generation of diversity. Well, we all know that science improves as we discover explanations that we put forward that are easier to reconcile with the evidence. And I put it to you that it would be so much more satisfying if we could find a representation that made life inevitable, an inevitable consequence of its intrinsic architecture rather than the unlikely probability that has to be offset by huge numbers. We need to find a way that makes life inevitable rather than unlikely. So how do we do this? Well, I don't know. I put the question to you, but I have some ideas about, I think, that should be really fertile for students to explore. I think more attention needs to be given to exploring the intrinsic architecture and dynamics of living systems to explain the diversity of life. And 4 areas look interesting to me. The concept of self-organization at multiple scales. Living systems clearly are self-organized. They have attributes where, you perturb them and they come back to a particular steady state. And that operates at multiple levels in the hierarchy. And what we have is these little nested systems of self-organization in larger systems of self-organization. Organisms, in some sense, are these hierarchically self-organized units that interact across scales as well as being autonomous within a scale. And there's a lot of work, theoretical work at the Max Planck on proteins in regard this way. I mean, proteins are very self-organized. They fold up. We'll talk about them in a minute. The other thing about living systems is that they're really robust. They're not like little pieces of engineering where you swap out one component and they break. They're incredibly fault tolerant. They're very robust. So the genotype can change quite a lot under the hood, but the phenotype stays the same. So you have all of this perturbation underneath the architecture, but the phenotype is resilient. And that has interesting implications for making evolution likely. And I'll talk about that soon. The third thing is modularity. Organisms are modular. You have different units that are fairly autonomous from one another. And that allows you to have mutations in one part of the genome that affect, say, the head, and animals sprout horns, but it doesn't necessarily have to affect the back legs, right? So you have autocorrelation, but you also have the separation of parts. Organisms are essentially modular structures, and each module is nested within one of these self-organization components. And then the last thing is dynamical systems. Dynamical systems have all of these properties: self-organization, robustness, modularity, and they generate novelty. And I'm going to talk about that a little bit. So these hierarchically self-organized dynamical systems are stable to perturbation, so they tend to be the same. But when you push them enough, they spill over into new configurations that are also self-organized, also robust, and stable. But they may have different arrangements of their component parts and have different phenotypes. It's a little bit like a landscape, like a ball. It rocks backwards and forwards in the valley. And if you push it up here, it rolls back down. If you push it up here, it rolls back down. If you push it enough, it might roll down to a new stable equilibrium and stay down here. This is a different stable configuration than this one. But within each one of these valleys, it's self-organized and robust. All right. So these attributes foster dynamic relationships in the genotype-phenotype map that make these systems behave in ways that are more interesting and innovative than we might otherwise expect. They behave not like things that humans engineer. They behave in interesting ways. This is a Julia set, and I like it because it sort of shows that you've got these, it's like a worm. These things are growing out of the screen and getting bigger. Other parts are receding, and your existence in a genotype is inside this tunnel. And you can imagine that if you're inside it, doors open and opportunities arise, and you move down those doors. And as you move through the landscape, the door shuts behind you. So constraints open up and they close depending on where you are in this particular landscape. So do we have examples of this? Absolutely. I'd like to Socratically ask people in the audience, but I can't pick on you and I can't see you. But this is an ovarian teratoma. This is a tumor from somebody's ovary, and it has teeth organized in a very organized way. It looks like a rounded jaw. It has skin. It has hair. It has muscle tissue. It's a different kind of stable structure. It's not a human at all, but it's got these self-organized components that are collectively modular. Here's another one. This is also a teratoma. You can see there's a set of molars. These look like molar teeth. There's hair here. There's skin on here. There's muscle. There's more teeth down here. These are perturbations to these biological dynamic systems. And these, of course, you know, very, very strange looking structures, and they're not self-replicating. But if these things were self-replicating, they could be like life forms. So these systems are very hard to model. There are too many temporal and spatial context dependencies. So it's very hard to make testable ideas to see how they work. So it's all right for me to speculate that this may be close to how life works, but how are we going to test it? Well, we can learn something by looking at simpler systems that aren't as complicated as organisms, but that have similar properties, such as proteins. This is a protein, and proteins sort of wobble. They're self-organized, as I mentioned before. They have helices and sheets. They're self-organized. They snap up. They have a string of amino acids. They self-organize to the same configuration most of the time. They're robust. You can have lots of mutations. Different people have got different sequences of hemoglobin, but their hemoglobin works just as well. So the genotype-phenotype robustness clause holds. They're modular. You've got different elements. You've got, helices and sheets and turns. So they have different components to them. And the mapping between the levels of organization is very well characterized. We have the genetic code from DNA to amino acids, from amino acids to helices and sheets. We have the particular sequence of amino acids. We can see if something's going to be a helix or a sheet. And then we also have a pretty good idea about how these helices and sheets fold up to form the tertiary levels of the protein that actually are the functional components that operate in organismal systems. So I'm going to use protein evolution to try to make the case that intrinsic architecture is important in shaping the trajectory of evolutionary change, not natural selection. It's an intrinsic property of the architecture of proteins that shapes their change. And I'm going to tell you that natural selection may not be as influential in generating the diversity in proteins as is often assumed. So to do this, I'm going to start with a very simple model, first put forward, a graphical model put forward by this man, John Maynard Smith, in 1972, before many of you students were born. This is an Englishman, and he was an aeronautical engineer that turned to evolutionary biology. And he proposed this idea that we could look at the proteins in a space to understand their evolution. And I'm going to put it to you very, very simply. Imagine that you've got a very simple protein. It's only made up of 2 amino acids. And at the first site, you could have one of 20 different amino acids here, A through Y. And here's the second site, and A through Y. So we've now got the entire space for a 2 amino acid protein. Any point on that space is going to define a particular string. So here's the 2 amino acid protein. It's site 1 is H and site 2 is leucine. So histidine and leucine. So we go to H, whereas H is there, and L. If we put a point there, that should be this particular protein. Oh, there it is, I guess. H and L. There we go. Let's look at a different one. Valine and lysine. VK. V here and K across there. All right, so this is very simple. Let's do something a little bit more interesting. Let's look at all proteins of length 3. Well, we just need another dimension, right? So we need a third dimension for the third protein. So Here is serine, histidine, methionine. So serine along this axis, histidine here, and methionine into the board. That point in the volume corresponds to that string. So you can see what we're doing here. To look at a real protein, which is maybe 100 amino acids long, we just need 100-dimensional space, which is hard for humans to imagine, but easy to represent statistically. All right. So now I want you to imagine an amino acid that's a protein that's 100 amino acids long, and I want you to have one mutation, so one of those amino acids changes to something else. So 99 of them are exactly the same, 99 axes are identical, and one axis is different. So in something called a Hamming space, they're adjacent neighbors. These things are adjacent neighbors. They've got 99 dimensions, are identical, and one is different. Well, we can map all viable one-step mutants from that original string, and we can see that they form a cluster. Each of these different amino acid strings is a different string. They're all closely related in this high-dimensional space, and that blob is all, let's say, hemoglobin. They all work. They're very robust, and they're neutral. They're equivalent. It's a neutral neighborhood of sequences that are equivalent. Now, the multi-dimensional shape of this set of points describes the neighborhood is characteristic for each protein. And it reflects the constraints and evolutionary opportunities associated with a protein. So I like to think of it as this high-dimensional weird shape, right? So this is a 4-dimensional shape projected into 2 dimensions. So, you can imagine that, Dr. Spock on Star Trek sees something like this and goes, oh, that's the opportunity space for I don't know, some kind of insulin, for example. That's the opportunity space for insulin. It's very characteristic of a protein. So it describes, all of this volume describes the space available for evolutionary sequence exploration. So the lineages of a molecular phylogeny of bits of DNA are exploring new sequence combinations within that space. And so we can estimate this very funky space by sampling sequences across species. Each of these different strings is a different dot in this space. And so all of these dots, if we let evolution run its course, they will describe the convex hull, join the outermost points together, will describe the volume of that shape. And a phylogenetic tree based on molecular data is simply the path of sequence evolution through that space. You don't know the direction from this, but if you knew which was the route, this is an exploration of sequence space by sequence strings. All right. Now we're going to use that analogy that I've just hopefully introduced you to look at connectivity. Different proteins occupy distinct neighborhoods in that protein space. So these are sort of blob-like things that I've just made up to kind of represent different kinds of evolutionary opportunities associated with a different protein. This might be hemoglobin, this might be insulin, this might be some other protein. They're all different, right? And they're in different parts of the space. But within each one of them, lots of different dots making up different strings that describe that protein space. So let's zoom in on the neighborhood for protein A. Here's protein A. This is made up of tens of thousands of points. Each of those points is a string. And what you can see is any point here is going to be a long way from the points over here. They're very different in that space. But look here. A sequence in this brown patch is actually a neighbor to the sequence in this black patch. And occasionally they're close enough that a single mutation can push you from one phenotype to another. These are entry point corridors. Well, this was described quite a long time ago. Actually, so there's the entry point. Martin Heinen showed this for RNAs, that you could flip from one kind of an RNA to another by mutations through these very, very closely associated strings that had very, very different phenotypes. So here is a sort of an animation of what we're talking about. Here's protein A. This is the volume, the sphere, the protein space for protein A. And here is the volume for protein B. And these are all different sequences in this volume. And I'm just going to show you. There's a little dot there. That's a string in protein A, right? And here's that little dot on the phenotype. So as we move through this space, there's another little dot. There's another little dot over here. So what you can see as I add dots, these are all different sequences that have the same phenotype and code for protein A. And you can see on the right in the graph, we have these points that are coming up here. Up to this point here. At this point, the genotype-phenotype map changes. We have a phase shift, and the next mutation is way over here. A tiny point change in the sequence space and a big change in the phenotype. This is a phase shift. This is where we're moving over one of these barriers. And then, as we continue, you can see that as we move, we're still in protein space B, and these show this kind of static behavior here. Well, this is all very well, but are there any examples? There are hundreds of examples, and I've just shown you some of the earlier ones. Malate dehydrogenase, certain variants of malate dehydrogenase can be converted to a different substrate completely, lactate, with a single residue. This was shown in 1988 when I was a grad student. Similarly, Dalal in Lynn Reagan's lab in 1997 at Yale showed that you can turn a beta sheet, this beautifully organized structure, into an alpha helix with a single amino acid change. Just wonderful. And we've now got ARC repressor genes, lots and lots of examples of single changes result in a huge transformation of the protein phenotype. So these phase shifts, these transitions are reminiscent of patterns that we see in the fossil record. But is there evidence for this type of resilience mapping at higher levels of organization? Absolutely there is. I'm going to tell you a really good story about a graduate student who's now a professor. He's a graduate student in 2000, George von Dassau, and he was trying to understand Drosophila development and model it mathematically. And it turns out that in a certain stage of development, Drosophila have this syncytium. There's a sort of ball, hollow ball of cells, right, shown here. And they go through this phase where they have these gene expression stripes associated with the segment polarity gene network. So all of these stripes are physically on the sort of surface. You can't see them, but in terms of gene expression, they're striped. And these are really fundamentally important for developing segments in insects. Well, we know quite a lot about the cells in these segment polarity gene network. And so if you see this little box here, I've expanded this box right here. And these are sort of a model of what the cells look like. And they're highly stereotypically organized. For example, engrailed, in this particular line here, is going to be no engrailed, yes engrailed, no, 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 yes, no, 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 yes. And then that same set of cells will have, wingless, off, on, off, 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 on, off, 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 on. And patched is off, on, off, on, off, on. We see this repeatedly. And so what George von Dassau wanted to do is find out how the hell that was regulated. How was that controlled? Because that stereotypic behavior would provide insights into how the segments were formed on insects. So turns out we knew quite a lot about the wiring of these various genes. And here's a wiring diagram. One on this here is for a single cell. And we see that engrailed, the RNA goes to engrail the protein, which inhibits cubitus interruptus and promotes hedgehog. And so we have this wiring diagram. And George took this wiring diagram and transposed it into every one of the cells in his simulation and had these elements diffuse across the cell junctions. And he wanted to find out what the concentrations of all of these different transcription factors would need to be to make this pattern. And I'll cut to the chase. He failed miserably. He explored billions of combinations on the computer and could not get it to work. After 6 months, his advisor, Gary Odell, said, "George, I shouldn't have let you do this project. It's too risky. Do something else." Well, George wasn't about to quit that easily. He'd spent a lot of time doing this. So he wanted to just get at the last bit. You know, he's a very stubborn. Stubbornness is a very good trait in scientists, and he's stubborn. And so he jumped on the lab phone, and this was before, you know, all these apps where you can call people around the world for free. And the lab had quite a big international phone bill, apparently. This is probably apocryphal, but this is what I'd heard. And he asked different people if the wiring diagram might need to be modified, if there was something wrong. And my understanding is that he called, I think it was Michael Ackham at Cambridge University, who said, "Well, yeah, it looks fine, but I've actually had a postdoc, and it looks as if there's feedback from wingless, the protein, to wingless, the RNA, and there's also inhibition from cubitus interruptus to engrailed." So George said, "Oh, thanks very much," went back, changed the wiring diagram, and reran his simulations, and the very first time he did it, exactly this pattern. Well, he couldn't believe it. He thought, I must have done something wrong. So he unplugged these two elements, the dotted lines, and it broke, didn't work again, plugged it back in, worked perfectly. So then he said, what is the chance of me getting this right the first time? It's incredibly small. So then he spent the next few weeks trying to find the parameters that would break it, that would not work. And what he found is that the weakest link was robust over 2 orders of magnitude. In other words, this wiring diagram working in all of these cells is incredibly robust over a lot of variation, 100-fold variation, being the weakest, most sensitive jujitsu point, if you will, of that network. So this is a very, very robust network, which is exactly what you want when you've got something as important as making segments in insects. So he explored 10 to the 5th different parameter values for networks similar to the one found in nature, and gave the correct expression. When they used the one found in nature, expression was robust over 2 orders of magnitude. An amazing piece of work, and was published in Nature. So now I'm speculating. That's George doing rigorous science. This is me, speculating. So I put it to you that developmental phase transitions associated with robust architectures, like the ones that George demonstrated, might account for punctuated changes in the fossil record. So as Arthur mentioned, I work on sharks. And right at the beginning, I showed you this curiosity. Where does this hammerhead come from, this lateral expansion of the cranium? Well, traditionally, we have, believed that, people look at some strange trait and say, what's that for? Well, people say, well, the hammer must be useful for something. And people say, well, what do you think it's used for? And some people say, well, the hammer expands sideways so that the eyes, which are perched on the top here, can give you very good peripheral vision. The hammerheads have got, 360-degree peripheral vision, not very good binocular vision, but good peripheral vision. And people say, well, this allows them to see their food, squid that they often feed on. So then other people say, no, I don't think it's that. I think it's that you have this lateral expansion is favored because you can put all these electrical receptors on it, which gives them much better triangulation. You spread out the electrical receptors, and they can identify prey far more easily. It gives them better triangulation. And somebody else says, no, I don't think it's that. I think it's because, if you put an hydrofoil on the tip of an organism, it gives it mechanical advantage, which lets them turn really quickly. And then somebody else sees one in an aquarium and it's launching itself into the mud. So he says, I don't think it's a hammer, it's a shovel. They're digging with it. So all of these views, crazy, perfectly reasonable, but also a little crazy ideas, stem from the idea that this hammer is useful. And the idea that there's been natural selection slowly increasing the hammer over time till you get this amazing thing in Eusphyra blocki, which is the winghead shark, which is a crazy animal. It's about 4 feet long, and the cephalofoil is about 3 feet across. It's like a swimming T-bar. It's crazy and it's nuts, but it exists. So people are trying to make the case that this evolution occurred and it is the crowning apex of natural selection. Well, we've done a lot of sequencing, and when we first looked at all of these hammerheads, we found a different result. This is what we found first of all when we looked at DNA sequence comparisons. We found that the winghead was right at the bottom. It was basal, and then if anything, there'd been a diminishing trend of the shapes of the skulls to the bonnet head. Well, of course, we got that result, and I thought, well, that must be wrong. It's probably some gene. So let's look at a different gene. Well, we looked at a bunch of different genes, and then we thought, and they got the same result. So we thought, okay, maybe those genes are linked. They're giving us a bum steer. So we then looked at hundreds of genes across the nuclear genome. They all give the same result. At a certain point, scientists collect evidence, and we have to believe the evidence. Our theories hold up with very little data, but when you have a lot of data, we have to abandon them. So I put it to you that what's going on here is this phylogeny that we've based on DNA sequences suggests that there's been a quantum change, some aberration that's gone in the developmental process where there's been unregulated growth of these structures laterally. And then, if anything, there's been natural selection against the head, against the expanded head. This tells you the power of phylogenies. The previous phylogeny that had been asserted just based on gradualism and looking at these animals roughly by phonetics suggested that natural selection was favoring a wider head. But the data based on the phylogeny based on DNA suggests exactly the opposite. This shows you how these trees can actually influence the development of ideas. Well, are there other examples of these quantum aberrations? So many. But evolutionary biologists like to explain them away as the consequence of natural selection. This is a larval fish, Gibberictes latifrons. These are the pelvic fins that have got these ridiculous appendages on them. And people say, oh, they're used for display, they're used for camouflage, they're used for this, that, and the other. Here's another one, Lepidoptera, a cave bass. Here's a little video of it. These things are ridiculous. This is a fish on its dorsal fin. It's got this structure, which slows it down. It's like a ball and chain. It's basically setting it up to be eaten. There are a lot of examples of these things, and evolutionary biologists always say, oh, I wonder what the adaptive advantage of that is. And they come up with lots of ostensibly credible ideas. Well, I want you to question them. I'm not sure that if, what would it take? What would it take for somebody not to believe that this was the result of natural selection, right? So I think that's probably a better way to view it. I suggest that these aberrations are caused by perturbations of self-organized dynamical architectures. They are not structures that are optimized by natural selection. They are living versions of these teratomas. They are peculiar consequences that are innovations that the teratomas, of course, don't replicate, but these things can replicate. So this is conjecture, not fact. I conjecture that most proteins and many genetic networks have been selected. Natural selection obviously happens, but I'm just not sure that it's as ubiquitous as my colleagues would have it. I think they've been selected on the basis of robustness, the genotype-phenotype robustness. That the design principles underlying cellular machinery involved in the development are genotype-phenotype robust. Like cell cycle is self-organized. It's an important thing. Robustness persists at inclusive levels of organization and occasionally induces these phase transitions associated with innovations and aberrations, right? All right. So that's the claim. Well, what are the consequences of robustness for evolvability? I told you that robustness actually has important consequences. Well, here we go. Robust many-to-one mapping, so you've got many different genotypes all mapping to the same phenotype in here. Each one of these are different sequences. These allow more of the genotypic space to be explored without a phenotypic consequence. There's no consequence. They don't change in phenotype. So the more robust the module, the better the connectivity in sequence space. So let's imagine that each of these is a different module. This is a green module here, that one phenotype. This is a purple phenotype. This is a blue phenotype. And this is a black phenotype. And this is a red phenotype. And this is a yellow phenotype. The more space these occupy, the greater the chance they will bump into one another. Here's one which is not robust at all. Here's the same yellow, red, black, blue, purple, and green. Well, in order to go from green to purple, you have to have about 25 mutations happen simultaneously so that you can jump there. Well, that's not going to happen. You're going to be stuck on this little island the whole time. But over here, you can meander around, and just occasionally, if you happen to be here and you mutate over there and the environmental conditions are right, you can survive over here. This provides stepping stones to go from one phenotype to another. So interestingly, robustness, actually, the genotype-phenotype buffering, this robustness fosters a capacitance for change. The nature of the connectivity of these groupings dictates the evolutionary lines of least resistance. In this case, you could have one that goes around like this, or you could have another one that goes through like that. These are the lines of least resistance that the architecture dictates are likely to happen. If things are fragile, well, it's pretty impossible to go from any one of these dots to anything else because you need 20 or 30 mutations to happen simultaneously to launch yourself from one spot to another. Hope you guys are with me. So discrete responses that are associated with these many-to-one mappings like this, these discrete responses here, this is a response, this is a response, this is a response, obviate the need for natural selection to evaluate these incremental changes. If one-to-one mapping between genotype, and you've got, I don't know, 27 different steps, natural selection has to look at all these intermediate values, has to have selection here, selection there, selection here. Each of these green arrows is an incident of selection. And to go from black to red, you have to go through all of these transitional stages, and natural selection has to be invoked all the time. With a robust architecture, sure, you have natural selection, but you could be neutral within this group and you have natural selection from here to here, neutral within this group, natural selection from here to here, and neutral within this group, and natural selection from here to here. So here we have 3 incidences of selection that evaluate the phenotype. Here, about 27. Selection is invoked, it's not necessary to invoke when we have these robust architectures, right? So in a changing environment, evolvable organisms are going to outcompete those that are less evolvable. So the capacity to evolve, this robustness, is going to induce evolvability. So it itself should be subject to selection. All right. So the 4th thing that I wanted to talk about was dynamical systems, right? So I just given you ideas. I want people to question things. Curiosity and questioning is the fundamental axiomatic start of all scientific disciplines, right? So we don't yet have a general theory that explains how perturbations interact with self-organized architectures to generate novel outcomes, right? Where do these damn colored pigeons come from, right? But we can get some insight by studying how self-organized patterns spontaneously arise in systems that are not living, that have homogeneously distributed material. You know, Brazilians have a tradition for liking the beach. So here's some beach pictures, right? All of you have seen these beautiful patterns that arise naturally. These are just self-organized patterns that you get just from wave action. You see these beautiful patterns arising just inertly. There's no living systems that are involved. We can also get insight from beer. So I don't know if people drink Guinness, but you may have noticed, I love to ask the undergrads this, the bubbles in Guinness go down. But people think bubbles are supposed to go up because they're lighter. So I ask the students how this can possibly happen. Well, the answer is It's not an optical illusion at all. It's real. The bubbles are going down. But what's happening is there's bigger bubbles that you cannot see in the middle that are going up. And because they have more force, they go to the middle. And there's lots of small bubbles, and they have no place to go. So the thrust of the big bubbles pushes the small bubbles out the way, and the small bubbles appear, well, they go down to the bottom of the Guinness. What you can't see is the big bubbles that are generating these vortices, right? So, all right. So in the same way, you can have this self-organized convection cells arising in weather, and they're called Bernard rolls, right? So if you get some fluid, some viscous fluid, and put it between two sheets of glass and heat the bottom piece of glass here, they will actually generate these regular sized vortices, these cells. And here's a little simulation showing exactly how this happens. You heat the glass at the bottom, and the heat rises, gets to the surface, cools down, and then becomes denser as it cools, and it naturally forms these cells. You can see that these cells is a diagram showing how these ones form. You can see these convection cells form naturally just with energy from the bottom. Well, this is what they look like from the top. You get these things forming from lava. Lava will form these pavement cells. What this is exactly this, but viewed at from the top. You can see these are moving, and these elements are changing shape, but they form these convection cells, which when they harden, form this cell-like matrix on the surface. All right. Well, Alan Turing was interested in how you get order from chaos. He was really interested in these self-organizing systems. And he showed prophetically that people didn't believe for 50 years that homogeneously mixed reactants can generate coherent spatial patterns. When the product of the reaction activates its own production with a positive feedback cycle, but only while activating the production of an inhibitor that diffuses through the system even faster. Well, here is a sort of a diagram showing the activation and the inhibition. This is the inhibitor, and they're auto-inhibiting and auto-activating here. So here's the auto-inhibition and auto-activation. This simple system generates beautiful patterns. And this was shown, here's the differential equation showing that, but I think students prefer to see an example. Well, this is the Belusoft-Zabatinsky reaction shown with these fluids. And Zabatinsky looked at this for 20 years and then eventually gave up, and then Belusoft came up because people didn't believe him. This is a homogeneous reactant showing products that organize themselves in these beautiful patterns, exactly mirroring what Alan Turing had predicted from first principles. You can get these exquisite patterns, and you can adjust the kinds of patterns that form by changing some of the parameter values in these dynamical models, the amount of inhibition, the amount of diffusion, and the amount of activation. Well, people had asserted, Turing asserted, that a lot of the patterns that we see in biology, the skin surfaces, were Turing patterns, and people didn't believe him. But now, after 50 years, people are looking and dissecting these things, and indeed, Turing was right. These are all Turing patterns that we see in nature. You can generate these with models. In fact, we described, Will White, Peter Last, and I described an Australian stingray from Northern Australia, and I wanted to call it Turing Eye, but we called it Australis. But this is a 2D model of reaction diffusion based on this set of inhibitions, and it looks really similar to the patterns on this particular stingray. So, and other people have shown that the stingray patterns are often consistent with Turing patterns. So these Turing processes have now been shown to underlie limb development. Small changes in the parameters can explain how limbs evolve from fins to limbs, allowing vertebrates to conquer the land. This was work that was done on a shark, Cilia rhinus, and mouse. And this was done by Onimaru, was published in Nature in 2016. And this is a limb bud of Ciliarinus. And the model predicts for these particular elements, BMP, SOX9, and Wnt, these patterns of expression for the Ciliarinus and this for the mouse. These are theoretical predictions based on particular parameter values. These are the observed expression patterns for SOX9 and SOX9 for the mouse. The mouse one is not exactly the same, but these are uncannily similar. And I agree with Onimaru et al. I think this is a beautiful piece of work showing that just by changing some of the parameter values, tiny little changes, you can change the expression of these genes, which causes cartilage condensation to go from a fin to a limb. So I suspect that these Turing-like processes are behind a lot of the morphogenetic sprouting that characterizes the diversification of life on Earth. So I think we can move from my childhood obsession with these just so stories to models based on Turing-like dynamics. There are others, not just Turing's, but others too. So I think we can get rid of these sort of magical ideas and replace them with scientific ones. So this is sort of philosophical way, because I've got about 5 minutes left. I think different ways of viewing life have different implications for how we view our place in the universe. As scientists, our philosophical perspective about our existence changes on the probabilities of processes happening around us. In other words, ideas have consequences, huge consequences sometimes. If the origin of life is an inevitable consequence of chemistry on planets that are similar size to Earth, we should expect absolutely to find life throughout the universe. And if that's the case, we should stop wondering if we're alone in the universe and instead start thinking of where to look for other forms of life. And NASA is doing this. I can never be sure of anything, but I'm confident that in the next 1,000 years, we're going to find evidence of life outside of our planet. And this is a little controversial. My colleagues don't like this. If speciation is just primarily a process brought about by architectural perturbation, as I'm suggesting might be the case, rather than a consequence of optimization to perfection, the traditional view, maybe species turnover, speciation and extinction, should be seen as ephemeral processes, not something that we need to freeze or conserve or freeze midstream just to maintain the status quo. So the whole idea of conserving things in a static way is like freezing a snowflake. Every snowflake is unique and ephemeral. If we let these processes, it's a natural part of it. So maybe what we're trying to do is freeze snowflakes as the dynamics of the globe changes. Of course, my colleagues hate that because they think it means that we can destroy or people will think that it means you can destroy the world. Well, that's not what I'm saying. I'm just saying that speciation and extinction may be natural processes that we should tolerate, right? These origin questions underpin religion and science. So one team has gone for magic, and it seems to be unusually popular, at least around here. And the other team has gone for Newton. And the Newton's team, so I myself have no expertise with magic. I'm just a boring scientist. But we don't have the answers. Newton's team does not have the answers. There is a lot of mystery that remains for those of us that are using Newton's approaches to explore and explain. So I think this is a wonderful time to be a scientist, to wonder about the architecture of living systems, to see if we can find a way that makes the diversification of life inevitable rather than unlikely. And so with that, I'll take any questions that anybody's got. Thank you.
Wow. Honestly, I gotta say that this was seriously one of the best lectures I have ever taken. Thank you so much, Mr. Naylor. I'm gonna send a link to one very dear friend. You probably know him. I wanted him to talk to you because he also has some interesting questions he wanted to ask you. But I also have questions. But thank you very much for your time, for your lecture. It was extremely good. I believe we have some questions on the chat. I'm gonna check now.
Okay.
Okay, but I just wanted to start with a question I have, and I believe this is like the first thing I wondered when I started my dissertation with phylogenetics and anatomy. I heard that we have a lot of molecular research going on to propose relationships and evolutionary tree lines in many different lives of forms: fish, sharks, beetles, whatever. But I believe the main challenge for that is to detect which gene is responsible for coding the character you're coding in your morphological analysis. In other words, you are, for example, coding the limb shape of a shark, or the fin shape of a shark, and the limb shape of a mouse, and you know that the gene associated to that is SOX9. So, has anyone ever tried to consider combining those things, like to say, okay, a limb coded in state 0 has this sequence on gene SOX, while another species has the state 1 has this sequence on SOX9. You know what I mean?
Yeah, I know exactly what you mean, and I don't think people have tried that, but I think that what we probably find, this is a guess. This is not. This I don't know the answer to your question, but my guess is that the gene sequence itself will not really map very well to the trait. I think the regulation of that gene, the transcription factors that switch that gene on and off, might be much more tightly associated with the trait. So, because the genes, the proteins themselves, can vary a lot, you can have tens of thousands of different sequences that code for the same SOX9 that does the same job. But if that SOX9 is upregulated or downregulated, then it becomes available for the animal to use, and the cells will respond to it, and you'll have cartilaginous condensation. So I think that the regulatory elements are likely to be more tightly correlated with the character states that you observe in the phenotype. That said, that's a speculation. I don't know if that's true. It could be the case.
Mm-hmm. Yeah, I also have another question, but I'm gonna save it for later because I believe this mysterious friend will be here soon. If anyone has a question in the chat, please feel free to ask. Snelo will be able to answer all of them. Se alguém quiser perguntar alguma coisa, por favor, fique à vontade, usem o chat, tá? Mario will be here anytime soon.
Good.
Okay. Before he comes, I guess I'll go with another question. You mentioned the tree of the hammerheads, and
Yeah.
this is very interesting because I mentioned before we started this lecture that one of my PhD ideas would be working with the cephalic anatomy of this family, which I believe is awesome. When you show that the Osphyra blocki is the aberration that actually worked out to generate new head shapes onto new species of hammerheads, I was very curious about it. Um, do you really believe that this is an aberration that really worked out, or do you believe there's, for example, an intermediate stage?
Yeah,
This
is very interesting because I mentioned before we started this lecture that
Mario, you have an echo. You have an echo. Oh, there you go. So as I was saying, do you believe that this is an aberration that worked, or do you believe there's some link species between the ancestral state of a flattened head, of a pointy head, between and this species and the winged head shark? You know what I mean?
Yeah, I don't think there are any intermediates. I
Yeah,
think
that's it.
that I think that I think that we're always looking for missing links, and I think sometimes we're looking for things that don't exist. My guess is that that these animals, there was a a polymorphism, and some of the animals had these very bizarre heads, and they probably could survive in suboptimal habitat, and they couldn't do as well as some of the other sharks, and so they stayed in this suboptimal habitat, and they're all in there together, polymorphically, and eventually, because they're sort of marginalized, they probably start breeding true. Of course, you can't just have one; you have to have a bunch of them, right? Before the trait becomes, established in a population.
Cool. Thank you for your answer. Mario, if you want to ask, that's your time. I believe you know him.
Hi, Mario.
Mario, your microphone is muted. Mario.
Hello there.
Hey, Mario.
Can you hear me?
Yep.
Absolutely.
Thank you very much, Gavin. That was a great talk.
Oh, thank you.
Really something that is, much broader and more interesting than boarding sharks. I'm wondering how to go into other subjects because, you ran out of sharks to study. Too few of them. So I'm really glad that you're into this kind of interest because these are questions that actually drive our curiosity in terms of biology and evolution and broader subjects. So your presentation was very convincing in terms of the structuralist basis for diversity and complexity.
Yep.
And I think it is quite interesting. I'm mostly convinced by it, or at least that the factors that you described and explained are important in the phenomena of life. However, the question was, why so many different forms of life? So my question is, considering everything you said, why diversity accumulates? Why we have so many things on top of one another living together? And why don't we just have a few forms of life that get increasingly complex because of structuralist reasoning and phenomena? So then I think we need additional factors like geography, like ecology, and other kinds of interaction that will result in diversity accumulating over time into this wonderful biosphere that we have. So what are your views on these things?
I think you're exactly right. So if you take my arguments, so the structuralist arguments, you would have increasing complexity localized, but you wouldn't have necessarily the layers of dynamics. But I think what happens this is a guess, speculation, is that as you have more of these animals arising, they create new habitats. Their existence intrinsically becomes habitat for others. And so essentially, I view it like if you're trying to fill a space, like a box, an empty box, if you put some big balls in it, then you might be able to put some smaller balls that will fit into the cracks in the big balls. And then you've got smaller cracks that you can put smaller balls into. And this approach generates more complexity and more opportunity for other organisms to eke out a living where they would not be able to. And we see that with ecology. Complex systems, you can't just, monoculture crops don't work as well as polyculture arrangements of lots of different organisms. Of course, you have to get the polyculture correct. But this is a speculation, but it's a point well taken. I think that the number of species is probably a consequence of interaction among species that generate new opportunities for other innovations to occur.
Yeah, I think that too. But the thing is, there's an additional factor, I think, which is geography. In order for things to actually have an opportunity to evolve independently and not be destroyed or incorporated by other systems, you need to be protected for a while. This is, of course, very similar to traditional speciation by isolation.
Yep.
This is something that goes back at least to Buffon. So the answer may be very, very old.
Yeah, no, absolutely. Certainly, if you separate things, they will not be able to interact with one another. And then if they become so differentiated that when they do come back together, then they've already made themselves discrete. So absolutely, isolation through geography is a way to multiply and compound the effects that might be intrinsic to the systems which are interacting organisms and the architecture which promotes dynamism. Absolutely, I think that's right.
Okay, just a quick note, Mr. Naylor, if you want to stop sharing your screen, that would be good because we can see like a bigger picture.
Okay.
Mario, thank you for your question. We have a bit of an echo, so if you can just mute your microphone, it would be good.
Yeah.
Thank you.
Okay.
Okay, so we have time to 2 more questions. We have a chat question now. Let me just see here. This is the question from Santiago Montealegre, and he says, "We tend to think that adaptations are for some purpose. For example, the horns on some ungulates. We think it is to defend ourselves. However, horses and zebras do not have them, and therefore we could think that the unicorn is impossible to have existed. My question would be, does your analysis support this idea?"
Gosh. So I think that the way to address that, I mean, so the honest answer is I don't know. But I think the way to imagine it is the opportunity space that might exist for unicorns, horses, ungulates, and the architectures that might be possible among those animals. I'm pretty sure you could make a unicorn if you took some of the tissue from an ungulate and you seeded it in the sagittal plane of a zebra, you might be able to induce it. But that's not really the same. That's a mechanical engineering kind of a operation. I see no reason that unicorns should not exist. But it might be hard to get to the unicorn space from horses or from ungulates, right? I mean, carnivores are closely related to horses. Perissodactyls are more closely related to carnivores than they are to ungulates. So, so I think the question is a good one, but it's one that I think that I cannot give an answer that is helpful. Do I think it's possible? Yes. Do I know how you would test it? No. And that's the problem with these dynamical systems, is that they are so complicated and context sensitive that it's hard to come up with tests that allow you to further basic knowledge about the systems.
Great.
I'm sorry if that's an inadequate answer. I realize it is, but it's a good question. I guess I don't know. I don't know.
I thought it was a pretty complex question, but I believe you answered pretty well. We're gonna have another question in a minute. I'd just like to get an opportunity to make another one. I mean, I have lots of questions. We'll talk later, but this one I also think it is pretty interesting to analyze. You mentioned it is about the winghead shark. It is a pretty rare species. It's not easy to find. It's not easy to see in collections. So I believe there is some sort of component that makes its viability difficult. Therefore, we see such a low number of specimens, not either captured or in collections. Do you believe that there's a correlation between what could be an aberration in anatomy and species rarity?
That's a great question. I don't know. So all I know is that the more diversity that I encounter, the more surprised I am that some things exist. And so just because a human thinks this is an aberration, it can't, possibly make a living very competitively. Maybe it can. And certainly, Eusphyra blocki, the winghead shark, used to be far more abundant. Its diminishment is largely associated with fishing pressure because they've got these big heads and they get caught in nets. And so they've suffered more than have other sharks. But I think that just because we see something that looks ridiculous doesn't mean that it shouldn't exist. Or that it should be rarer. I mean, one of the things that I just do not understand is why Lepidoptera go through a larval stage, which is just eating. They then go into a chrysalis, which is basically a really nice protein for a predator that doesn't fight back, and then go into a reproductive stage like a butterfly or a moth. It seems ridiculous that you would go to such levels of change. You reorganize with the imaginal disc all of your biochemistry, reorganize your cell structure, and come out as a butterfly. I mean, if there were no butterflies, might be a possible life form, I would say that's ridiculous. Well, guess what? They're incredibly successful. The Lepidoptera have been tremendously successful all over the planet. They're crop pests. They're huge numbers. And so this is a strategy that I would not think was an enduring one, that would not last very well, but I would be really, really wrong. So, I think you have to be careful about anthropomorphizing and saying that looks like a crazy structure. So then, I can criticize myself and say, Gavin, the reason these larval fishes with these long appendages, just because you can't imagine how they would be useful, you need to get inside the head of the fish. Maybe they are useful, right? So that's what a selectionist would say. So I think we have to be careful about asserting what is useful and what is not. But I think that the reason these wingheads have gone down in number is largely because of fishing pressure. They get caught in nets a lot.
Yeah, same rules apply for Isaac and for them, unfortunately, because I was wondering about my target species for the master dissertation, but I believe that when you mentioned the three of this furnace, I believe that spider block eye would be a better example. We have a comment in the chat, and I'm waiting for the question that Mario will send on the chat. I believe it's Mario's cousin. It's from
Cool.
Marcel. It is from Marcelo Fukuda. Fantastic talk. Thank you very much. It's not a question, but rather a comment. It is always good to remember how humble we should be in interpreting the things we see in nature, or else we always have the risk of being overconfident and become truly believers in our stories and forget to keep asking questions.
Yeah, I 100% agree with that. Absolutely.
I also agree with that. Uh, I'm not sure if we have a lot of time. Okay. Okay. So he had to leave. Okay. So I believe we're done. And Marcelo says, thanks for bringing such fantastic examples. Reminds us of this. So once again, thank you for Marcelo, and thank you from the organization committee. A huge thank you for myself, Mr. Gavin. It was truly amazing to see this lecture. It was truly amazing to see how complex our life is. And thank you very much once again. You're always welcome to join us anytime you want.
Okay, thank you very much. It was a pleasure. Yeah, I think there's always more questions than answers, and I have lots of questions and very few answers. So this is a privilege for me to talk to you all.
The privilege is always. Thank you. Thank you very much. Okay, so thank you all for listening to us on the audience. Muito obrigado a todos que participaram. Em breve a palestra vai estar disponível. E é isso aí, meus amigos. Uma boa noite. Thank you all. Good night. Just a second. I believe the transmission will be over soon.